All right, we're about to kick off the next talk in this room by our very guest, Martin Larralde. I butchered the French name. But anyhow, he comes from Paris and he lives in Leiden now. And he worked on research and he also did a lot of C++ work, C++ work, low-level stuff, touching lots of legacy code. How did that feel? Are you fine?
It's really hard. It looks horrible, but yeah, you have to make it work.
Yeah. Cool. And you are gonna tell us a few things about DNA, and you take it from there.
Okay. Well, thank you so much. Yeah. So this is a bit of a change because usually I present at scientific conferences, so I can't really nerd about the code, but I guess now I can, so that's really exciting. However, I still have to show you a bit of biology so you know what's going on before bombarding you with some Rust. So like, bear with me for a second, you're gonna have to do some genomics with me. And in particular, I mean, I'm interested in the study of like a couple of proteins that are named transcription factors. And these proteins, they can bind DNA at a particular site. And this binding, it's probabilistic. It's not really a yes-no answer. It has some energetic constraints. So it's like maybe it binds here more than there. That's how you need to take this into account. But once that protein binds, then it expresses a particular gene that is located after that site. So we really want to, when we have a big sequence of DNA, we want to figure out where these sites are located and where these proteins are going to bind so that we can figure which genes are located after them. The problem is that these transcription factors, they don't have just one binding site. As I said, it was probabilistic. So all we can actually observe is a bunch of validated binding sites. This we know by experiment that this particular protein from this particular bacteria is going to bind to these sites. But from that, we can build a consensus and kind of have an idea of, yeah, okay, that protein recognizes more or less this sequence. But once again, it's probabilistic. So it's not like you can just apply an exact sequence, like string matching algorithm. You can't just write a regular expression for this or like do hey-ho Korasik or whatever. So the question is, how do you find new potential binding sites for a known motif for which we have experimental data in a new sequence? And here it's a sequence that just comes out of the sequencer. It turns out the answer to that problem is actually older than me. It was proposed in 1993, and like that's kind of still something that is being used. And the idea behind it is to take the binding sites that have been experimentally validated and generate a count matrix. So you count at every position of that site the occurrence of the different symbols. Here it's DNA, so you have 4 symbols, ACGT. The original paper is for proteins, so that's generic over the protein, the alphabet. We'll come back to that later. But from that count matrix, you can generate a scoring matrix. Once again, it has statistical implications. I'm not covering them. It's just a way to score a particular symbol at a particular position. That's why it's called position-specific scoring matrix. How do you, with this scoring matrix, find new potential candidate sites? Well, you take your scoring matrix, you take your candidate site, and you just read the score that corresponds to the symbol at each position. So here the candidate site starts with a C, so I take the C score, then I take the T score, then I take the G score, and so on. I get all my scores, I sum them, I get a match score. This score, once again, has some implications, but we don't care about it. All I care is to compute these a lot, because I'm going to compute this for the entirety of the DNA sequences that I'm processing. So I need that to be fast. And as a sort of benchmark, I wanted to look at implementations that are out there. And to do that, I made a small benchmark with just like one cute bacteria, Escherichia coli, that is everywhere, and just like one single motif of 16 symbols that's sort of in the median range. And I took 3 Python packages, but before you flame me, all of them are under the hood in C. So like, it's a fair comparison to relook at performance for these. And what you can see is that the performance is that, we are in like megabytes per second. I mean, it's not too bad. And okay, sure, you can see that the fastest one, it takes about 200 milliseconds to compute for the entire sequence. So here maybe you're like, okay, you're over-optimizing something, but you need to take into account the genomic scale. Here I'm showing a bacteria. Bacteria have small genomes. If we want to look at a slightly more like larger organism, pluricellular organism, like completely randomly a crab, I don't know, then you actually have a genome that is 1,000 times larger. Here we're talking gigabytes of DNA. And usually you don't want to scan with just one motif, you want to scan with a database of motifs that are known. So this database is also large. That means that here, that's 6 orders of magnitude more computation than you need to do. So I did the math for you. That's over 11 days of computation. And that's really a waste of your tax money, because I would do that on a public cluster paid by your taxes. You can parallelize that. Sure, it's embarrassingly parallel. I could do multi-threading on my computer. That would still take a day. That's not ideal. And the reason it's not ideal is because actually the implementation for that is not super complicated. I mean, here it is. It's like, yeah, it's two nested for loops. And for every sequence position I'm going to score, I compute a score. And that score means for every row of my scoring matrix, I add the score for that symbol. That's actually a good learning exercise for bioinformatics students. You can really give that to them. And so here, indeed, what I did was I wrote it in Rust, but I'm hiding a bit of the complexity in the different types. We come to that later. One thing you can see here is that, yeah, the runtime complexity is OML, where M is like the number of symbols in my matrix and L the length of my sequence. That's not going to change. I'm not doing some mathematics algorithmic optimization. I just make the OML run faster. So we can really quickly talk about biological alphabet representation, because one thing I said was that the method actually works for both DNA and protein. And another thing is, you don't really want to keep your sequence as a string, because a string, even if you do an ASCII string, well, it has a lot of characters that should not happen in DNA, usually. I mean, DNA, it's 4, okay, maybe 5 characters if we have a wildcard. So let's have an enum for that. Let's write the nucleotide enum, and here I'm using rep U8, so like my enum is a bit smaller in memory. And I'm also explicitly putting the numbers. I'm not sure you need to do that, but that's just me being paranoid. And finally, I have like a default, which is the N, which is my wildcard in DNA. And I could stop here, but I'm just going to add a tiny bit more of abstraction by having a trait that corresponds to a biological alphabet. And this trait has also an associated symbol type, which also implements the symbol trait. So maybe naming not ideal here, but like whatever. And also a constant k, which is the number of symbols in my alphabet. And I can implement that for DNA. Sure, now DNA is like a marker type, but that's going to help me for generating generic code. And then I could also define that for proteins, but I'm not showing you. And that means my sequence now, instead of being like a vector of bytes or a string, it's a vector of alphabet symbols. So I'm generic over the alphabet in my sequence type. And I'm also generic over the alphabet in my scoring matrix, but for now it doesn't really come into account. It's just really used as a marker. I'm saying this scoring matrix was made for that alphabet. This means that now our scoring code, which had this signature, turns into generic code over the alphabet. So this has 3 results. One, we have the same code for DNA and proteins, which is like great, because actually that's not always the case. We have sequence validation in advance. So that means when you are processing data, we know that already we only have like the 5 symbols we care about. And we ensure the scoring matrix and the sequence are in the same alphabet, which is also sometimes a mistake, which is not great. And so how does Rust compare actually on our old benchmark? And that's really the Rust implementation I showed, plus these couple of extra traits. Wow, it's so fast. I didn't do anything, and it's like already 10 times faster than C and C++. And so, yeah, congrats compiler team, I guess. I mean, I don't know who to thank, but like, thank you. Yeah, that's really great. I mean, because we took like an existing algorithm and just porting it in Rust and doing a bit of clever type declaration and so on seemed to have made it faster. And all the rest I'm going to do now is just added bonus. This added bonus is going to come in the form of AVX2 instructions. And here I'm getting into platform-specific code. So in particular, I'm going to use this gather instruction, which is an instruction that when you have a base, like some data located at a base address and an index vector can be used to load multiple elements from that address using the indices from that vector. And so this you can do in Rust using like the SIMD intrinsics. My idea behind it was to use it to, at this stage when we are basically summing over the different symbols, to do something a bit like this. That's not valid, but like the idea would be, okay, can we get the scores for multiple symbols at the same time and compute multiple scores at the same time? So you can, but it needs a bit of like a funky sequence memory reconfiguration that I'm going to go over very quickly because you do not have to care about it. But the idea is that we take our sequence and we put it in a column major order matrix. And this matrix has a number of rows which correspond to our SIMD vector. Once again, not super important. And then to score, what we can do is that now we can load an entire row of our matrix, and we can use our gather instructions to get multiple scores. And then we can compute scores for multiple positions at the same time. And in the end, we also get our final score matrix in a column major order matrix. That goes into there. I'm not showing the code. If you know SIMD, it looks exactly the way you assume. If you don't know, then you better don't look at it. One thing you notice, however, in this algorithm is that we have matrices that have a constant number of columns, because here K is a constant that depends on my biological alphabet, and W is the width of my SIMD vector. So both things are constant, which means I can make my constant dimension matrix. I could use the N algebra crate, for instance, but I don't need algebra, so I'm just going to do it myself. And boom, yeah, I just define a row that is under the hood an array of constant size, and I define a matrix as a vector of this row. So it has like cache locality, everything. And then, well, that's my alphabet. So now I can make my scoring matrix a matrix that is where the number of columns in that matrix depend on the alphabet. Or at least I would love to do that when that gets stabilized. So currently, if you try to compile this, you get an error, but I'm sure if you go to this other talk, you will get some information about that. Very nice. So for now, I'm stuck using the generic array crate. That lets me do basically the same thing, except I can't really use constant. I still need to encode my numbers as types, but that's fine. I can still do what I want to do. I can still get a scoring matrix that has a constant number of columns. So how do we compare now that we have SIMD? Yay! Well, now we are 2 orders of magnitude faster. So indeed, I can process gigabytes of DNA per second. 1.1 is gigabytes. And I didn't lie in my talk. So that's already like super good, because now that means, yeah, this analysis I was talking earlier about like looking at a crab genome, now I can do it in half an hour. Cool. And probably less if I parallelize. The problem is that that's me. I'm excited about Rust. Bioinformaticians are not there yet. You know, their favorite programming language is still Perl. So that's where we are. You know. So if you want to make stuff useful, you need to make it available in a language they figure out. And for that, you can do Python bindings. And once there, I'm not gonna really go into detail because I mean, as everybody is using, we're using Py03. It's amazing. Go to the workshop if you want to learn more, I guess. Finally, I'm running a bit short on time, but I just wanted to see SIMD in Rust is so much more easier than in C. And it's easier for several reasons. First one is memory alignment. In SIMD, you usually need memory alignment. In C, you need to use either non-portable functions, attribute, or you need to do manual pointer arithmetics, and you end up with like these kind of things in your code. In Rust, you can just say, please align my type. Yay! So once again, a good thing from the compiler. Conditional compilation, well, Same for C, we need to do a lot of things at build time to check, can we compile for a particular architecture, particular features? You need to write header guards everywhere. In Rust, okay, I have a couple of like conditional compilation things, but I can force the compiler to always emit AVX2 code, and that's not a thing you can easily do in C. And the final thing that is much easier in C, in Rust and in C, is dynamic dispatch. And here, I mean, in C, you either have to use an external library, which means extra dependencies. You need to read from like a platform-specific location. You need to use the CPU ID instruction to figure what are your current CPU features. In Rust, there is a macro for that in the standard library. So yes, thank you. So in conclusion, Rust for biology, I mean, I think it really has a lot of potential, in particular for people that are currently doing C and C++, and that has already started. There are like a couple of very high-profile tools that probably 5 years ago would have been written in C or C++, and that have been written in Rust, and now people are actively using them. But also for things like portable code, it's actually more important than you think in biology, when we have like clusters that are old and people that are running on Windows. Reproducible builds, dependency management, and so on. I will just say the ecosystem is not there yet. We have a BioCrate. Fun fact, it's actually the first open source Rust project I contributed to in 2016. But it really has not evolved that much. I can say that there is a lot that can be done in there that it's a bit lacking compared to, I don't know, BioPython, for instance. So there is really room for improvement, but the ecosystem is a bit behind. And finally, yeah, SIMD in Rust, so much easier. It's unsafe, so you need to do what you're doing. But I guess STD SIMD is coming eventually, and that will be occasion for a great rewrite. So thank you for your attention.
Thank you very much, Martin, for your talk. Also, thanks for saving taxpayers' money with Rust. Want to move to Germany?
I moved out of Germany, so but I was wasting your money already, so don't worry.
That would be another talk. We don't have time for that discussion today, but we have time for a few questions. So if anyone has a question, raise your hand. It's hard to see people up here. Yeah, there is one here. Yeah.
Do you have any idea why the C implementations are so much slower? Because if they're slower even for a single-threaded execution, then it's not just less parallelism. Are they just written poorly? Are they not utilizing SIMD? But even your implementation without SIMD was much faster. Do you have any idea what's happening there?
That's a good question. So they are not using SIMD, but even without SIMD, we are faster. So that's that's using the super optimized SIMD version. I tried to look at the BioPython one, which is not the worst one, but it's the one that is like at 10 megabytes per second. And I can't really say, because it's not that different from the one I showed earlier, like our Rust version, the native one. What it just does differently is that where I'm using a lookup table, it's using a switch. Is that the reason? Can really a switch degrade your performance that bad? I'm not sure. So maybe that's that, but that's the only thing that really looks different. Otherwise, it's still using like a dense matrix. It's still using the same kind of code, let's say. So I don't know.
More questions?
In the front.
Hi, thank you for the talk. You mentioned using AVX2 to accelerate. Have you looked at AVX 512?
I have not, because the Leiden University cluster is running on CPUs that are already 10 years old. So even AVX2, I have to explicitly request an AVX2 node if I want it. So AVX 512, yes, it's the future. I think that's not the most urgent thing for us in bioinformatics right now.
Thanks.
There's one more question in front.
One of the big problems in bioinformatics right now is multiple sequence alignment, and what you've showed is using one genome. But if we have multiple genomes and we're trying to, say, build a tree to see where a certain species originated from, would it be trivial to expand this method to multiple sequence alignment?
I mean, this one is clearly made for single sequence. In a way, it's like it's a way to do ungapped, pairwise, local sequence alignment. For multiple sequence alignments, you have other options. I mean, there are tons of methods basically developed. If Rust can help, I don't know, probably, because currently, I guess a couple of them are written in C, C++, whatever. The problem is always adoption of these methods. Like if you make a new one and you want scientists to get it, you need to properly benchmark and show, oh yeah, it's producing the same results, or at least results that are better on a biological point of view. So that's always a bit hard to do when you come from more like engineering background, because all of a sudden you're like, okay. And otherwise, you need to make a tool that does exactly what the other tools are doing. That's less interesting, but that's another option. And then you can convince people to use it just because it's faster. But here, I really went for tools that do simple stuff. I'm not trying to like build on a new method. It's really exactly a method that already exists. I just show to people that you don't have to wait for so long until it gives you a result, basically.
Right next to you, on the left side. I guess she also had
Hello. So I believe the scientific world is more like there's two paths of optimization. Like A is like with the Rust parallelization, like GPU, CMD, like more like system level of optimization. Another approach is like just make an efficient algorithm itself. So like I'm like having a hard time to prioritize, like, because actually mathematical optimization gives much more performance, but it takes longer time. So to make that algorithm to be stable and then like make it working. But the system approach have a limitation on like how many limit that I can optimize on, but it's very easy to like add on it. So I'm just wondering like what's your decisions or like a time allocation, resource allocation on this thing? And just like a small question, like have you also considered like GPU optimization apart from SIMD? Thank you.
Yep, thanks. That's a good question. I will say probably for the nice mathematical optimization, there is maybe a bit less knowledge in that area. Period. When people are developing things, even the people that are developing high performance methods do not necessarily have like that background in bioinformatics. So that's actually something that can be harder, and that's why a lot of people just kind of do the heavy crunching. Can we just go faster based on that? Another reason is a lot of optimized, or by that I mean fast software we use in bioinformatics, they tend to be built on heuristics much more than like a nice, elegant optimization to make them run faster. And these heuristics are like a super big trade-off and a super big question about what you generate. Is it biologically relevant or not? And then people start getting into faith about what kind of parameters you need to use, because then it's like, okay, if I tune down this, it will run faster, but I may miss some things. So it's really hard when you have these heuristics. So that's why for me, I tend to just go like the systems optimization approach, where I just look at old software and sometimes I see, okay, I mean, this part has been really coded like shit, but like we maybe just by fixing that, we get a bit of improvement. Sometimes it's 100 times, sometimes it's like 2, but like that's already better than nothing. Yeah.
Fantastic. Give it up for Martin. Thanks for your talk.